Now let's get started with our presenters. First, we have Matthew Albrecht, the conservation scientist at the Missouri Botanical Garden. Matthew.
Good, thank you, and I really appreciate the invitation to come talk with you today about the Center for Plant Conservation's Best Reintroduction Practice Guidelines. I'd like to first acknowledge my co-author, Dr. Joyce Mashinsky, who's with us today. Joyce is actually the lead author on the CPC Reintroduction Guidelines, which were published back in 2012 in a book called Plant Reintroduction in a Changing Climate, which you can find on Island Press's website. I highly recommend you go purchase it for about 40 bucks. It's a wonderful book. And so I wanted to sort of take a step back before I talk specifically about the guidelines and kind of talk about why do we need best practice guidelines for reintroduction. And what I'm showing you here are some slides from some work we've been doing looking at the role of ex situ conservation and reintroduction on endangered species recovery plans. These are federally listed species threatened or endangered. This is an analysis we did back in 2012, 2013. And what we found were that 77% of recovery plans for listed species include reintroduction as a recovery action or task in their recovery plan. And that's what I'm showing you up here in the left-hand corner, and I'm breaking it down by region. Here in the southeastern region, about 81% of those recovery plans include reintroduction as an action or activity to recover that species. In Hawaii, it's almost all species are going to require reintroduction to ensure their persistence in the wild. That's a whole nother ball of wax in Hawaii that is for another talk another day. But I want to point you over here to this upper right-hand panel. And what this is telling us is that over time, we're increasingly using reintroduction as a tool to recover species on the Endangered Species Act. So those species that have been listed more recently on the ESA are more likely to have reintroduction as one of their recovery actions. And down here in these bottom panels, I'm zooming in on the southeastern region and sort of asking under what circumstances are we using reintroduction. And we're using it for species that we tend to think have a low recovery potential in the wild. And when we're using reintroduction and recovery plans, we're using them as a secondary or tertiary activity. In other words, it's not the first thing we're doing when we're recovering a species, but it's a secondary or tertiary activity. And that's consistent with how we view reintroduction in the CPC guidelines. If I could use this word during an election season, reintroduction should not trump other primary conservation activities, such as habitat protection, habitat conservation, habitat management, discovery of new populations. Those are the first things that we typically do in a recovery plan, and those should precede a reintroduction. We should also add that reintroduction is really something that you first have to have good ex situ conservation practices in place, and that's what the last capacity building session was about. You need to be able to control the threats and understand the threats to the species to have a successful reintroduction program. These are all things you have to consider when you're deciding whether or not to proceed with the reintroduction of an endangered plant. And if you'd like to learn more about that, there's some great text and boxes in the CPC guidelines that I would highly encourage you to read if you're thinking about developing a reintroduction program for a rare species. So our goals for reintroduction are to increase the spatial occupancy of the species on the landscape and to increase that species abundance to reduce its extinction risk. And it's really something that is designed, and this is how the guidelines are designed, to be done in tandem, integrated with ex situ conservation and a variety of other activities, including habitat management, habitat restoration, of course, to get to our ultimate goal, which is to ensure that rare plant species continue evolution in their natural context. So if you look at the guidelines in the book, they're basically structured into 5-part sections, beginning from what I just talked about. Should you even be doing a reintroduction for the rare species? What types of species should we be doing? Justify a reintroduction, all the way to monitoring and reporting those reintroductions. And what I'm going to talk about today, because I only have about 15 or 20 minutes to talk about the guidelines, I can't possibly talk about all aspects of the guidelines in that short a time. So what I'm going to talk about is the sort of preparation phase of a reintroduction program, because we think that's one of the most important parts. And then the folks following me are going to really talk about implementation. We're going to get some case studies from the southeastern US from Mike Kuntz. We're going to get some, we're going to have a talk on monitoring and reporting, and then we'll also have a talk on managing reintroduced populations in the wild. So really the first step when you're developing a rare plant reintroduction program is to develop a reintroduction plan. And we think that that's critical to the success of a reintroduction program, a very well thought out plan. And in some ways, a plan should really be one of the most time-consuming things you do in a reintroduction, is developing a very strategic plan. And the guidelines, I think, are really well written in the sense that they give us checklists of things we should be thinking about including in our reintroduction plans. And I'm giving you an example of one of those checklists there. There are a myriad of factors you have to consider, from what genetic material you're using, site conditions, site management, and basic things you need in place in terms of aftercare and long-term monitoring to ensure success of your project. So a real theme in the CPC guidelines is that you have to understand the underlying biology and ecology of the species that you're reintroducing in order to have a successful reintroduction program. And I put sort of three, what I think are key factors, but there certainly are many more that could be put up here. But it's really important to know something about mating system, because that tells us something about how we should maybe structure spatially our reintroduced populations. That's also related to the genetics as well, which I'll talk about in a moment. But we also need to understand the interactions between a species and its abiotic and biotic environment to have a successful reintroduction program. And finally, we need to understand something about the habitat or microhabitat preferences of that species. And this last part is something that we've been thinking a lot about in our reintroduction programs with rare plants in the Southeast. And something we've come to realize is that many of our rare species, it's difficult to tell what the evolutionary environment was for that species in the modern landscape. And so many of the rare species that we see existing on the landscape exist as remnant populations, and many of them exist in degraded habitats where the processes are no longer operating that help that species to persist. And so many of our current populations are actually demographic sinks. They're actually on a slow trajectory of decline. And so going out and using the habitat of the remaining populations of a species and trying to reintroduce a species to sites that mimic those conditions is likely going to set you up for failure, because it's likely the conditions that these rare species exist in are actually degraded and suboptimal. Let me give you an example. This is Pine's ground plum, Astragalus bibulatus. It's federally endangered. We began a reintroduction program with it back in 2000. And at the time, this species was known from just a handful of populations on the edges of limestone cedar glades in the Nashville Basin. And this was a species that at the time we thought required semi-shade at the edges of these glades. So the reintroduction program was structured around reintroducing the species to unoccupied glades at the edges. And after about 10 years, all those populations went extinct. And we then quickly realized, after doing some demographic work in the natural populations and relating that to the microenvironment, that this species is actually threatened by woody encroachment, and its growth and reproduction fitness is actually maximized in open environments. So it turns out the ecotones around these glades have cedar encroachment and woody vegetation encroachment that really threatens the species. But at the time, we didn't really think that. We thought that this was actually suitable habitat for the species. So now we know that this is what this species needs. It needs fire to open up the woody encroachment on the edges of these ecotones to provide more optimal habitat conditions for the species. So when you're preparing for a reintroduction, there are really three dimensions when you're designing a reintroduced population that you have to consider. Your genetic dimension, your horticultural dimension, which was what the first capacity building session this afternoon was about. I'm not going to talk about that further. And then there's a demographic dimension. And these sort of all three interconnect and overlap. And I'm going to spend the rest of my time talking about the genetic and demographic dimensions. So as a restoration practitioner and you're doing a restoration, one of the first things you have to consider is what genetic source material are you going to use for your reintroduction or restoration program. And a big thing in the restoration literature is, should you be using single population genetic sources, or should you be mixing your population genetic sources together? And so traditional paradigm tells us that really local is best. We shouldn't be mixing our population sources together in restoration programs, because we could be disrupting locally co-adapted gene complexes, and we risk outbreeding depression when we bring genetically different sources together. They cross and they have offspring that have lower fitness than their parents. That's been the traditional paradigm. But for many rare species, more recent literature is suggesting that maybe that's not the case, that in the modern landscape with habitat fragmentation, we need to maybe be rethinking about restoring gene flow on the landscape and bringing populations together. So this is a really great example of this from Joyce's work down in Florida with the endangered beach cluster vine, which is a perennial vine endemic to coastal dunes. And what she did was they had reintroduction sites scattered across the coast here of Florida. This is the east coast of Florida. And at these reintroduction sites, they planted individuals that were derived from different pollination crosses. And it turns out that those individuals that were crossed from different distant genetic sources actually had higher fitness at the reintroduction sites than those that were crossed from local sources. And it turns out that over time, the reintroduced populations from mixed population sources were actually more resilient to stochastic disturbances. In this case, it was a hurricane. And so this is an example where sort of reconnecting gene flow was really important for this species to build a more genetically resilient population. So in many cases, we don't have genetic data at the beginning of our reintroduction program, and we certainly often don't have what Joyce had, which were all these wonderfully crossed plants from previous research to reintroduce into our site. However, there's been some great new tools for us to use to make decisions about when we should be bringing different population genetics together. This is a really important paper published back in 2011 in Conservation Biology. It's a decision tree that you go through and answer these questions in this decision tree to determine whether or not you should be bringing your genetic sources together for your restoration program, or should you be keeping them separate. Essentially, if you have clearly resolved taxonomies, you don't have fixed chromosomal differences among your populations, and you think that in recent ecological time there has been some gene flow, then it's likely by bringing sources together, different population sources together, you're not going to have outbreeding depression. And there are several other guidelines and decision trees that have been developed, and I have those listed on a literature cited section that you can find on one of the tables in the back here. Okay, so those are the sort of genetic issues you have to deal with. Then there are the demographic issues, and this is really a question I get asked a lot for reintroductions. How many propagules should we be reintroducing of a rare plant to an unoccupied site to create a viable population? And really, it depends. This is some work from Tiffany Knight in the same book that the guidelines were published, where she basically asked the question, what are the minimum number of individual seeds or transplants do I need to create a viable population of a rare plant? And we all know, if we're working with rare plants, that our germplasm is very precious and in short supply, so it's important we maximize our germplasm in these restoration projects. So what she found is that species that have a great deal of year-to-year variation in their demography, they fluctuate wildly, you're probably going to need a larger minimum founder size to create a viable population. Species from stable environments that have very sort of low year-to-year fluctuations, you typically can use a smaller founder size. That's what I'm showing you on the left panel. On the right panel here, it also depends on the habitat. If you think that you've optimally matched the species niche to the habitat, then you can probably get by with a smaller founder size. If you think that you have a suboptimal habitat, you're probably going to need a larger founder size. And so this goes back to this idea that you really need to understand the habitat of a species. And one thing we advocate in the guidelines is to use experiments to test the habitat preferences of rare species during your reintroductions. This is an example from Astragalus bibulatus. Again, these are ecotones at the edges of these limestone cedar glades that have become mesophyed over time from woody vegetation encroachment, a process called mesophication. And so what we did was we hypothesized that if we could find more open xeric environments, that the reintroduced transplants would actually perform better than they would in a more mesic environment. And that's what we found over time, and we structured this as an experiment across this ecological gradient at multiple sites. And another thing we emphasize in the guidelines is that without experimentation, that you can sometimes draw wrong conclusions. And you really don't learn anything if you don't set up your reintroductions as experiments. So this is an example from the same reintroduction where we're looking at the demographic traits of reintroduced astragalus plants across this ecological gradient. What we did is at each one of these different habitats, we caged half the plants and left half the plants uncaged. So had we not done that, we would have concluded that none of the sites could actually support the species because only one plant or two plants flowered over a course of 4 years, and that would not be considered a viable reintroduction. However, it's not the abiotic environment that can't support flowering plants. It's actually the herbivores that are preventing certain plants from flowering. And so if we look in our caged plot of our more optimal xeric habitat, we had 28 plants flower over the course of 4 years. And so this is an example where using experiments, we can sort of disentangle the role of abiotic and biotic factors and understanding what sort of drives the persistence of reintroduced populations. Another question we ask in reintroductions is what propagule stage we should be using, seeds or transplants. And we know from some work we did for the reintroduction book that if we use larger propagule stages, we have a higher probability of that reintroduced population persisting over a longer time scale. Why? Because we basically have skipped that demographic bottleneck that characterizes so many plant species, seed to seedling life history transition. You can skip that and you can have a higher probability of persistence. But again, it sort of depends and interacts with the propagule stage and life history of the species. This is some work from Tiffany Knight did in the book as well, with some modeling work of the demography of natural plant populations. And really what she found was that if you look at a gradient from short-lived species to long-lived species, it really changes your minimum founder size you need to create a viable population. So for very long-lived species, trees, long-lived perennials, you're going to need a lot of seeds to create a viable population that's going to persist over long time scales. Why? Because seeds have very low reproductive values for perennial plants. In other words, the probability of that seed ever becoming a reproductive plant is extremely small. That's less so the case for species with shorter lifespans. And in some cases, you may need to decide if you're working with something like an annual, that the trade-off between using seeds and transplants, it may not be worth the cost to actually grow things out and transplant them into the wild. You may be able to get by with using seeds. In general, though, we know that using transplants, you're going to have higher success rates, and that's why the guidelines, really, we kind of came up with this 50 rule that we need a minimum of 50 individuals to create a viable population in our rare plant reintroductions. So finally, this sort of summarizes the preparation and the design of reintroduced populations. And I think one thing we can learn from our past work is that reintroductions really require careful planning and should be executed as experiments to gain more information about the species as we go. And we think developing a reintroduction plan is really critical to the success of many reintroduction programs. Thank you.
Thank you. Next, we have Mike Coons, a conservation ecologist from the North Carolina Botanical Gardens. Thanks, Mike.
All right. This is a talk. I'm kind of co-giving this with Wade Wall, who's here as well, and a whole bunch of other people you can see listed there. Matt Homan, who works with Wade at the Army Corps of Engineers, Johnny Randall, and Janet Gray, who's a botanist with the Department of Defense. And I'm going to kind of start to build on some of what Matt was talking about in his talk, looking at reintroductions, and we are kind of looking more at the implementation side, which I'll touch on a little bit, but also looking at that monitoring and some ways to kind of evaluate your reintroductions. So, as Matt was saying, this is a pretty young science, so a lot of this stuff that we're doing is very new. And we have a lot to learn from it. And I'll just kind of reiterate what Eve was saying. When you're doing these, we really need to be setting them up as experiments. There's a lot of very Johnny Appleseed approaches of just throwing some plant seeds out there and saying, hey, look, we did something good. But we're not learning anything from those. And when they fail, or if they are wildly successful, we don't necessarily know why. And we'll be able to evaluate that. So the project we're working on is this cooperative and collaborative project with the Army Corps of Engineers and the Department of Defense, which is in the Sandhills region of North Carolina. So here's the map here. Here's down here, tiny, Sandhills region, blown up. Fort Bragg is this large base right here, and small acres, training land, so very wild, very large landscape. It is a fire maintained ecosystem, longleaf pine savanna. There we go. We chose for this project, 5 different species that grow in different habitats across the ecosystem. We chose them because they grow in different habitats, so we got a wide range life cycles, life histories. Today we're really going to talk about these two, Amorpha georgiana and Astragalus moshoii. These are some of the ones that we've got some of the better data on and are looking at right now. But these are all species that we are still kind of working our way through analysis on. So the larger project, the objective to establish self-sustaining populations, viable populations. That's kind of the goal of most of these reintroduction projects. And to identify life stages that are critical to the positive population. And then also something to look at the cost analysis of these reintroductions. Thank you. I'm getting instructions to move a little closer to the podium. So in order to do this, we looked at all 5 of these species, and we outplanted different age cohorts at 4 different sites. Fort Bragg has really great data on all of these species, so we knew populations and local sites that had been. So we can target those for reintroduction. All of our outplanting occurred in the fall, and we did this over multiple years. Something that we have seen that we didn't, I'm not going to show you the data for today, but these different years have different success rates. The outplants we put out in 2012 have different success rates than the ones in 2013 and the ones in 2014. So, you know, these years matter. There could be factors in there that we don't understand that we need to evaluate further. For all of the reintroductions, we collected demographic data on these populations. For all phases of that. Down here at the bottom in this chart, these are the 2 species we were working with, and you can see, you know, Matt was just talking about the founder size individuals, and over those 3 years, the numbers of individuals that we outplanted at our sites. And then we did this in 3 different individuals, and their link that back to the. All right, so what is reintroduction defined? First, 96, and he talked about, and that's a really important, and knowing that you can project goals, reintroduce. Biologically. Be able to look at those reintroductions and decide if they were successful or not. A lot of reintroductions in the past have gotten kind of differing evaluations of their success. Godefroy did the different approaches and success rates for reintroductions. But most of these approaches are just looking at very simple metrics. Presence, absence, did the plants reach reproductive maturity? Are there recruits? Don't look at things over the long term. Garant in 2013 wrote a wonderful paper where he kind of looks at all these different metrics and says that we need some way to evaluate multivariate standards. Was to use comparative demography. So in our work at Fort Bragg, we wanted to look at how reintroductions compare to natural populations. We can create these demographic models, but if we don't know if they are doing better or worse than natural populations, we need a way to evaluate that. There's a few examples in literature of reintroduced populations being compared to natural populations, but it's something that isn't done extremely widely yet, and I'm hoping I can change more people's minds and more people will start looking at things in this way. Demographic data is great to use because it's a defensible metric. It's really a great tool to be able to set up evaluations. And then you can get into these comparisons and look at adaptive management, adaptive reintroduction protocols. So basically saying, let's find a way that we can learn from what we've been doing. So specifically, for our demographic methods, we marked all the individuals that were reintroduced individually. Each one got a dog tag. We can trace it back if we want to or need to at some point, all the way back to its maternal line, so the parent and the population that individual came from. And then we've done similar things in the natural populations, gone out and surveyed a variety of natural populations and tagged and flagged all the individuals. And then we go in and we measure every year survivorship and mortality, stem height, stem diameters. For the amorpha, we measure the crown diameter, flowering, fruiting, and number of capsules and astragalus. The natural population data set was actually built off of some work that Wade had started when he was in graduate school. So there's 9 years of data now, or is it 10? Yeah. So quite a high number of years. And then we can break all that demographic data down, and we created size classes for the two species, small individuals, small, medium, medium, large, and extra large, with some various sizes there for the different species. And these are all based on stem heights. All right, so what can we learn from that and what can we see? So here are some survivorship tables by size class, with the reintroduced populations being in red and the natural populations there in blue. This does not include the data from the year of transplant through the first growing season. So it takes out survivorship of those founder events and then looks at the survivorship from those dates forward. And you can see for the amorpha over here on your left, in most of the size classes, the small, medium, the medium, and the large, most of the survivorship from those individuals from year 1 to year 2, et cetera, is pretty comparable. Small individuals, though, in the reintroduced populations have a much lower survivorship rate than do the other ones. With astragalus, the story is very different, though. All of our individuals, we don't have any reintroduced individuals that have made it into the larger size classes, and none of our smaller size classes are having the same type of survivorship that our natural populations have. So one of the reasons to look at this comparative demography is if you were just looking at these survivorship bars for these size classes, you might think that you still have survivorship and that things are going swimmingly, but compared to the natural populations as a baseline, we know that that's not necessarily the case. And we've got to be able to figure out what's going on there, which I don't have an answer to today. But the other thing that we can look at then is if we take our size classes and we look at how much they're growing from year 1 to year 2, something that's pretty interesting to note is in the amorpha, if you remember, this is the small size category, and they were the ones that had lower survivorship. But the individuals that are surviving are actually growing at a rate that's greater than the natural populations. And the others then are all very comparable too. And the astragalus kind of tells the same story. Not only is there low survivorship of those individuals, but they're also not growing. They're not transitioning into those larger size classes at a quick enough rate, and they're staying small, which is likely going to increase their mortality. So Wade put together then also some transition matrices, which we'll just go through quickly. There's a lot of numbers on those. But you're just looking at how many of the individuals, a proportion of individuals that are transitioning from year to year to these different size classes for both the reintroduced populations and the natural populations. And for Amorpha, those transitions aren't all that different. They're staying fairly similar across those. And again, then mortality here at the bottom. You can see we haven't had any seedlings in the reintroduced populations where you do get seedlings in the natural populations, but there's really high mortality of those seedlings. So that's an important life stage and something that we need to, I think just haven't had enough time in the reintroduced populations to be able to identify that yet. We do have some individuals flowering, but haven't had any new recruitment in those to date. And then this is just a matrix that's showing then basically the most important size classes here in bold, which are a little hard to see, but the medium and large and the few extra large individuals there are really important. But those are the individuals that have reached a reproductive maturity. They're able to respond to fire and come back and flower and reproduce pretty regularly. Astragalus, again, this kind of goes back to that same story from those early graphs. In this transition matrix, we're looking at how many individuals are moving between these different size classes. And again, you've got from year to year, you've got a lot of individuals that are staying in these small classes and not any that are transitioning into the larger size class as of yet. And this may be a function of time, but it may be a function of something else that's going on ecologically at these sites. And again, this is the elasticity matrix, and looking, it's these small to medium and medium-sized individuals that we really need to make sure that we have into our populations. So, what did we learn from this, and where can we go from this, with this information? I've hopefully shown that comparative demography can play a really important role and can be a really useful tool in your reintroductions. It can give some insights into long-term success. I think from looking at these, we can say that the Amorpha populations that we've reintroduced have a much higher possibility of reaching successful populations or successful reintroduction than the Astragalus does by comparing them to the natural populations. And we've identified the life history stages that are important. Those small to medium size classes, we really need to find ways to get individuals in our reintroduced populations into those life stages, and whether that's starting with them, larger individuals that are already in those classes, or finding out if they just need some sort of additional aftercare to be able to get them into those places where they are more likely to survive. The modeling can provide insights into this long-term success. We've talked a little bit about that. And then this idea of adaptive reintroduction techniques. As we've done this once now, and we've had multiple events, but I think we've started to learn more things. These key features and these key components, these life stages that we need to be able to make sure that we can get into our reintroductions. And if you haven't seen this paper yet by Eric Manges, it's really worth checking out. And with that, just like to thank our funders and all the people who have contributed to that. Thank you.
Thank you. Are there any questions for Mike? All right, I saw 3 hands go up. That's probably all we're going to have time for.
Did you notice any insect herbivory on the Astragalus during fruiting? And I wonder if maybe that was happening to some of your small plants too. And if so, did you try any caging?
We have not caged any Astragalus individuals, but we do notice both herbivory on the plants and frugivory on the developing fruits. We get them both. Do you guys see that down here as well? Yeah. There's pretty low reproductive potential in Astragalus. They flower copiously, but there's really low fruit production. and not really high viable seed production within those. You do get viable seed out of them, but it's, if you saw one in full flower, you would never guess how few seeds you can get off of that by comparison. So.
Hey Mike, how much does root rhizosphere microorganisms have to do with the establishment of these introduced species, such as like mycorrhizal and other unknowns?
We didn't test any of that, but I imagine that has something to do with it. Astragalus grows in a really xeric sandhill scrub, so it's very possible that it has a lot of associations that we just don't know about. It could also be just nutrient deficiency. They grow, the habitat sometimes is defined as a pea swale, which can have higher nutrients than the other sandhills. That's all a relative term. They're still really depauperate soils. But it could have been a habitat, not quite finding that right habitat for the reintroductions, even though we were planting at sites that had previously had plants known at them. But I think microorganisms are something that need more attention and reintroductions.
I saw him near the front.
I can talk to you more later.
So you set that up. You had 3 different ages that you planted, and I was expecting you to compare the effectiveness of that. Is that in the works?
Yes, that's also in the works. And we are preliminary data, but we are seeing difference in survivorship between in that founder event and the survivorship. And correct me if I'm wrong, Wade, but I think as time goes on, because of those growth rates, especially like in Amorpha, those growth rates of the individuals are very quickly matching what you see in the natural population. So of the individuals that survive outplanting, they very quickly fall into that transition matrix that you'd expect. Now, of the individuals that survived outplanting, there's definitely a skewed curve towards the older plants surviving better. That's a whole nother level of data that we can break down to explore.
Thank you so much, Mike. Our next speaker is Carrie Radcliffe, the restoration coordinator here at the Atlanta Botanical Gardens. Carrie.
Thank you, Gretchen. And I must say, I'm one of very many people who coordinate safeguarding and restoration efforts here at the garden and within the Georgia Plant Conservation Alliance. Many of them are here this week. So I'm happy to be here presenting on behalf of Atlanta Botanical Garden today, but I'm also acting as a mountain bog project coordinator and safeguarding database manager for the GPCA. We're going to learn a lot more about them on Thursday, but they've been around for over 20 years now, and they act as a collaborative alliance, umbrella alliance, to coordinate research, management, education, safeguarding, and recovery work in the state of Georgia. And their headquarters is at the State Botanical Garden of Georgia, but we work together as a cooperative, collaborative group. Here is a list of safeguarding organizations. Highlighted in red are some of the core horticultural operations that contribute to ex situ and in situ safeguarding work that we do, but it's not limited to these 4. And at the heart of our safeguarding program is the ex situ and in situ safeguarding components. So we are cultivating plant material from known wild populations. We're crosswalking those to our partners' data at Georgia DNR and their natural heritage database. This comes in the forms of plant, seed, and other germplasm, and serves as an insurance policy against catastrophic events that could occur in the wilds. I will say that our augmentation reintroduction programs focus on habitat restoration and field recovery, and that is of utmost importance. We're always working on managing populations, whether they're purely wild, augmented, experimental, or reintroduced and reestablished. That's what we're focused on here, is management and recovery goals. And all of our activities support this. So mountain bog safeguarding is GPCA's signature project. It's been going on since the organization was formed in the mid-90s. And mountain bogs are one of the rarest habitats in Georgia, as well as in the southern Appalachian Mountains. It is a very high priority in Georgia State Wildlife Action Plan. and they contain suites of bog endemic species that are restricted to one or very few sites. And so we're at the southern tip of the range for a lot of these species and have just one of some of them. So they're very, very important to us. I definitely want to credit the Conservation Horticulture Program for GPCA and at Atlanta Botanical Garden as revolving largely around Ron Dieterman, who was one of the founding fathers of the GPCA, getting that program up and running here before we even had a separate science and conservation department, and that they have been involved in all of these aspects here, including ex situ and in situ work. We propagate many plants for in situ recovery work here at the garden and at other gardens. There's a lot of things that go into this, and we meticulously keep track of all the data associated with these things, reintroduction being one of those end goals. So our mountain bog safeguarding sites are primarily located on the 2 northeastern districts of the Chattahoochee-Oconee National Forest here in our state. And we currently have 19 safeguarding sites here. So 8 of these already had these bog endemic plants occurring on them. 13 of these sites are outplanting sites, and you can see they range from federal, state, private lands, including also TNC. And then we have some other managed sites where we don't necessarily have the rare plant elements there or reintroduced there, but we're managing it because it is priority habitat, and all of those factor in with a GPCA cooperative planning. So here's a list of our ex situ safeguarded mountain bog endemic plants. We have 13 species at 4 different safeguarding facilities that are part of the GPCA network. 9 of those species have now been outplanted in situ through augmentations and reintroductions. And this is a small sampling of the overall total 100 plants across all habitats that are safeguarded ex situ by GPCA, and 51 total reintroduced back into native habitats. Mountain purple pitcher plant is the poster child here. It was one of the original species that the formation of the GPCA and the Mountain Bog Safeguarding Program revolved around. So this has been proposed as a candidate for federal listing by the CBD, and it is endangered and tracked by the state of Georgia. We have one extant wild population on the Chattahoochee National Forest. We did have a site on private property that's been extirpated in the past I'd say about 15 years. But we now have 5 in situ safeguarding sites where we've put that back into suitable habitat on protected land. And over the course of about 21 years, we've outplanted 687 individuals out into the wild. Now, this doesn't seem like a big number, but we're big on trying things out in small batches until we get it right and adapting as we go. And that's helped us have higher survival rates.